So my name is Waqar. I'm working with Dr. James Chinebal uh, in University of Nebraska Lincoln. So today I would like to discuss about like how quantitative genetics uh, affect the photosynthetic variation in maize. So first of all, I would like to talk about the photosynthesis. What is the photosynthesis? So you already know, like all the guys, like it is the well-known process which uses the light energy and it converted into chemical energy, like using the carbon dioxide, and it produces the oxygen and the food. So it is the source of oxygen uh, for like the for the whole world, and it is also so it is the so it also forms the base of the food chain. So let's discuss about why photosynthesis is hard to study. So like photosynthesis is really a complex process. So there are a lot of multiple biochemical and regulatory pathways are involved in photosynthesis. So there are a lot of genes which are involved. So this is a process also which so it is a very dynamic process, like which changes according with the environmental conditions. Uh, and so on, so there are a lot of other environmental factors like soil, water availability. So they all make the photosynthesis a really complex process. So why field studies are more important uh, in the photosynthesis, but most of the studies are in the controlled conditions, but we employed the photosynthetic studies in the field conditions. So field conditions provide like natural conditions, like so you can evaluate more effectively biotic and abiotic stresses under the field conditions. So, and there you can also evaluate the genotype into environment interaction in the field conditions, which is the base of the breeding. And you can also evaluate more effectively the selection, so it can so there will be a more yield stability. But the challenges are in the field conditions, so environment is highly variable. So there is a noisy data, and there are also technical limitations. So going in the field and managing the taking the data is not easy, like due to the weather and all the other stuff. So we employed a multi-spec. So this is a device which combines the 3 meters, like chlorophyll, uh, and the fluorometer and the spectrometer. So it measures the photosynthetic traits based on the two parameters. Like it measures based on the fluorescence, which like photosystem 2, non-photochemical quenching, and it also measures the parameters based on the absorbance, like VH plus, GH plus. So, and it also measures the environmental data, like relative humidity, ambient temperature. So it is also feasible, when we analyze the data so it is also feasible that time. So here is the picture of the data collection which were collected from my lab members like Dr. Marcin and Dr. Saha in 2020. So the source of the genotypes which I used for the analysis, so it is the Wisconsin Diversity Maize Panel. So it, I think, includes like 900 genotypes. So I used 750 genotypes from there. So it is a highly genetic, and so there is a lot of phenotypic diversity due to the wild relatives, land races, and so there is a large population size. So that definitely there will be a more statistical power. So it is also well adapted to the temperate environments, in Nebraska. So we also have 11.8 million dense genetic markers. So there will be a high chance of detecting the genes with high resolution. So here is the methodology which I used for my analysis. So first of all, discuss about the experiment design. So we used the 752 genotypes in two blocks of RCBD, and so there were 840 plots, totaling 1680 plots in the field. So then the data were collected, like I already mentioned, with the multi-spec. So we targeted 14 photosynthetic traits, and like the relative chlorophyll, photosynthetic efficiency of the photosystem 2, non-photochemical quenching, like that. So after that, the third step is like the data curation. So after the removal of the extreme values, we targeted 710 genotypes. And then we estimated the variance components like fitting the model in the LME4 package uh, in the R. So like using the plot level values. And then I estimated the pre-spats repeatability. Like, so pre-spats mean like before the spatial correction. So it like, so just averaging the plot level values and like estimating the repeatability. So post-spats repeatability includes like, so there is a package for spatial correction in R. So I use that by modeling like plot IDs as a random effect and light intensity as a fixed effect. So which is a environmental factor, and day of time as a random effect. So I estimated the blobs, and then again I used LMB4 to estimate the repeatability. So in the same manner, I estimated the blues, which are the best linear unbiased estimates, which I used for the GWAS analysis further. So here I use the genotypes as fixed effects, replacing the plot IDs. So the next step is GWAS. So I use the phenotypic file, which I mentioned already, 710 genotypes. And the genotypic file here is the 4.7 million independent SNPs out of the 11.8, which were the total SNPs after filtering the minor allele frequency, heterozygosity filters, some like stuff like that. So we use the form CPU algorithm to run the GWAS, and the threshold was the standard Bonferroni correction, which is mentioned here. Then I performed the LD analysis. So we use the Plink and put the 50 kb upstream and downstream interval on both sides of the GWAS signals. And we performed the EQTL GWAS. Then for EQTL GWAS, we used the 693 genotypes of the gene expression data, which my earlier lab mate, Dr. Torres. So he he performed that analysis. And so we used here MLM algorithm and the same data, like the same threshold Bonferroni correction. So here I want to present like how light intensity is changing across the time of data collection. So you can see like at the different dates, so there is a different, and there is also during the time of the day, there is a lot of variation in the field for the light intensity. So it shows like how the environments affect the photosynthesis process. So here is like the distribution of the billows, like which I calculated through the spats, and they are showing the normal distribution, so which I use for GWAS. So here's so this is the result. These are the results actually. So you can see in the panel A. So this is the pre-spats repeatability, which is before the spatial correction with extremely low repeatability values, and panel C is showing like the post-spats repeatability, and there is a significant improvement in the repeatability values, like especially for the photosystem 2, like 0.53 and phi NPQ with 0.52. And the panel B is showing here like the variance contribution of the different factors like genotypes, raw, column, light intensity, like how much they are contributing to the overall photosynthesis variability. So in this figure, actually, here are the results of the GWAS. So on the x-axis, there is the genomic. So these are the chromosomes, like showing the genomic position of the GWAS signals. And the y-axis is showing the RMIP value, which is a criteria which measures the, in the form CPU, how frequently the significant GWAS hit is repeated in the multiple iterations. So we use the iterations in the form CPU, like we used 100 iterations. So here are the results, like you can see above the 0.2 threshold, there are 6 GWAS signals labeled with the different letters. And so these are the signals for relative chlorophyll, photosystem 2, and so on. So here in the panel B actually is showing, so So panel B to G, they are explaining the more detail of the significant GWAS signals. So panel B is, if we discuss about here, so panel B is showing the blue circle GWAS hit, which is for the photosystem 2. And so the red circles, so they are showing the LD with that GWAS hit. So the LD threshold is here on the y-axis is 0.6. And so the x-axis is showing the genomic position. So same here, like from panel B to D, panel B to G. So we are discussing about how the different GWAS signals so they are in LD with different SNPs nearby. So here in this figure, so there is you can see like RMIP GWAS hit with red labeled triangle. And so there are 4 genes within the genomic region of this GWAS signal, which is for the relative chlorophyll. And so the red label gene is the main gene which we detected through the GWAS analysis for the relative chlorophyll. So the blue circles, so they are showing the they are associated with the genetic variation in the relative chlorophyll. So they are showing in LD also with the equatorial hit here, which is on the on the right side of the axis, like the log 10 p value. So you can see so they are showing the LD with that SNP. So it means so they are regulating the expression of the relative chlorophyll here. So, and if we talk about the panel B, C, and D, so here in panel B, relative chlorophyll is showing the significant difference between 2 genotypes like AA and GG. So which is at this SNP which we detected for the relative chlorophyll. So and the panel C is showing the significant difference for absolute chlorophyll, and panel D is showing for the difference between for the TPM value, which is a measure of the gene expression. So it means like all the both genotypes are on the x-axis, so they are differing significantly for these 3 parameters. So it means especially we talk about panel D, so it shows so it further confirms the role of the EQTL. So this signal, so this genomic position for the variation in the relative chlorophyll. So here are some of the genes which we found for the which we found in our GWAS analysis. So if we talk about for the photosystem 2, so they are involved in phosphorus homeostasis, and and for the relative chlorophyll, so it perform roles in the iron sulfur protein binding, and so on. So here, so after identification of the genes or detecting the genes in the GWAS hits, so we are performing the validation of that genes in the orthologs of the Arabidopsis mutants. So we ordered the mutants from the Stark Institute in California, and we are validating them. So right now we are going through the process of genotyping, and yeah. So after that we have a plan to take the phenotypic measurements using the multi-spec. So we are growing them in the controlled conditions in the growth chamber, and we will take the measurements using the multi-spec, and we will see the changes in the mutants if there are some changes after the knockouts of the gene. So if we talk about the discussion, so when we compare our study with the previous studies, so so you can see the previous study 1, so they used 256 common accessions genotypes, and the second study used 225, and with the 4 and 8, and so they targeted the 4 and 8 photosynthetic traits. So in our case, we targeted 710 genotypes with 14 photosynthetic traits. And if we talk about the repeatability, so the common traits are here: photosystem 2, 5NPQ and Q, and 5NO. And so the repeatabilities you can see in study 1 and study 2 are significantly lower than our. Our have 0.53, 52, and 0.31. So they are significantly higher than their studies. So one more thing, so our study, how it advances the analysis, so we address the environmental factors like the light intensity successfully day of collection, and so we saw the improvement in the repeatability results, and there were a strong GWAS signals like with the high RMIP value. So we used a 2D spatial correction for our to enhance the efficiency of our analysis. So key findings were there like so there were 6 strong GWAS signals, like so they have one relative chlorophyll hit. So one signal for relative chlorophyll, 2 signal for 5 photosystem 2, and 2 for 5 NPQ and 1 for QL. So yeah. So if we talk about the conclusion, like so photosynthesis is like essential for plant food production. So here we studied how the genetic variation affects the photosynthesis, and so there are some previous studies. So they reported. So they are mostly like in the controlled conditions. So they are with like some mixed outcomes. So in our study, we performed these analyses in like in the field conditions, and so we identified very promising genetic variants which regulates the photosynthesis mechanism. So how we can like extend or improve these analysis? So we can extend this analysis to multi-environment, like including multi-years, or might be the multi-locations. So it enhances also so we can enhance our understanding of the photosynthesis related genetic variation by performing more analysis. So yeah, so like so let's end about the talk about with the acknowledgement. So these are my lab members, like Dr. James Schnabel, and so the other members. So here my lab is, so we are harvesting the corn, I think in Missouri Valley last year. So thank you.
We have time for a couple of questions for me. One right here, Eddie.
I'm curious if and whether you accounted for the variation for the different multi-spec devices, or if that was absorbed in the spatial variation somehow.
So yeah, so we used different multi-spec, like I think so 5 to 6 multi-specs, and we adjusted the that variation like by adjusting using the light intensity, so which is the major source of the variation. So we adjusted that in the spatial correction. Yeah.
Question about I didn't understand fully how did you correct your data for the different light intensity, since we know that photosynthesis will respond not in the straight line manner, but there will be hyperbolic answer of photosynthesis to the light. How did you correct for that in your results? And the second question is also how long your measurements, for how long they were taken. Did you correct also for diurnal changes in photochemistry?
So yeah, so we took the data from 9 to 2 p.m., 9 a.m. to 2 p.m., like on different dates, like I mentioned, July 23rd, July, so like, yeah, in a July month on different dates. And we are trying our best to eliminate the effects of the light intensity by using different spatial analysis techniques like we use. So yeah.
But it's not about where you are in the field. It's about the light intensity, which is in the time of measure, since the multispec will put the light inside the cuvette exactly which was measured before you close the cuvette. So I don't understand how you correct for that in your results before doing the GWAS. It's really nice results that you got so many hits, which are related to photosynthesis, but I'm a little bit skeptical if they really, if they are not confounded by the light intensity and the diurnal changes in photochemistry.
I'm not sure about that, sorry.
Okay, very well. Let's thank Ali.